Hello and welcome to video 5 and the last video of chapter 17, Blood, for 2402 lecture. So we'll start off with, we left off with platelets, so now we'll talk about what platelets do. And what platelets do is called hemostasis. Again, here's a word to break down for you. Hemostasis just means clotting really, but hemo is blood. Stasis is stabilize or remain the same. So stabilize your blood, keep it from leaking out. It is a fast, localized, highly regulated response, which means that you want to stop the bleeding quickly. You don't want to lose much of your blood, right? I mean, it's your go juice. If you lose like a significant portion of it, you'll just pass out and or fall off your bike or whatever's wrong with you. Localized, you don't want it to start clotting everywhere. You just want it to clot where the leak is, right? Picture systematic or systemic clotting, not good. And of course, it has to be regulated, as you don't want it just going off all willy-nilly. Now there's 4 steps that are listed by your book. I'd have probably put in 6, but I'm going to keep it the same. And here they are. Vascular spasm. So you get a cut, first of all. Vasoconstriction is going to result, which basically it's like stepping on a garden hose. If your garden hose is leaking and you step on it, it slows the flow. So you constrict smooth muscle in the area, which allows the blood to be staunched a long enough time for a clot to form. And the first step in the clot forming is called a platelet plug, where the platelets become sticky and they actually just gum up the hole. They actually form a clog like in your sink. They produce and release chemicals which make other platelets in the area sticky as well. Then coagulation. Coagulation is basically solidification of the material. So the blood and platelets in the area are going to become a solid thing, which is the beginnings of a scab. Now there's a couple of chemical names here, and I've reduced it tremendously. It's really a far more complex procedure than I've listed here. But you have a molecule called prothrombin activator. So prothrombin, anytime you see pro, it means before. So prothrombin activator is something that's going to activate prothrombin. Ta-da! Well, prothrombin activator forms by this complicated reaction set, which I'm not going to describe. And prothrombin activator activates prothrombin, which prothrombin is a plasma protein. So you have prothrombin circulating around in your blood right now. If you get a cut, prothrombin activator forms in that area, which activates prothrombin and converts it to thrombin. Follow me here. Thrombin, then, the new molecule that forms, acts as an enzyme which catalyzes another plasma protein, fibrinogen, which is circulating around right now. Thrombin, once formed from prothrombin, converts fibrinogen to fibrin. And fibrin has a name which you can picture here. Fibrin, fibers. So fibrin is an insoluble filamentous protein which will then be laid down on that platelet plug and form within that platelet plug, forming this big gooey mesh of platelets with fibers running in between it. So you form this really kind of solid clot, which is basically a scab. Dry it out and it's a scab. So the last step here involves that sort of drying out bit, healing, and then getting rid of the clot. So it's really 3 things in one, which is why I wanted another 2 steps, but I'm, like I said, keeping it the same as your book. So the 4th step is clot retraction, healing, and fibrinolysis. And they all make sense. Clot retraction means kind of tightening up that knot. You kind of tighten the screws on it. The fibrin mesh retracts and kind of pulls the edges of that wound together. The platelet plug dries out and becomes more of a hardened scab. And therefore, now you've got a little tough cap on that injury, and you can all picture a scab. You start healing at this point. A clot and a scab is not new skin. New skin has to form, so you undergo mitosis in the area, fix the blood vessel, make new skin, etc. And at very last, you'll then do fibrinolysis, and there's that lysis word again, right? Fibrinolysis, breaking down the fiber. You're going to dissolve that mesh, you're going to dissolve that scab. The scab will fall off, and the molecule that executes that is called plasmin. So you've got plasmin circulating around. If it encounters a fibrous scab, it starts to work on that, dissolving it over days. All right, more on clotting disorders. There's a couple, 3 of them, I think. So thromboembolic disorders are a big class of things that include undesirable clotting in undesirable locations, for instance. A thrombus is just a clot that forms in the wrong place. If you've ever heard of deep vein thrombosis, that is the formation of a clot in a large vein, usually deep in your tissues, often due to inactivity. Say if you're already infirm and you go on a long plane flight for 15 hours, sitting there for 15 hours is not the greatest thing for your circulation, theoretically, and often you do form a thrombus. If that thrombus comes loose, it's called an embolus. And when that embolus reaches a smaller tunnel than its diameter, it's going to get plugged. So you may have heard of a pulmonary embolism, lung clot block. If you've got that clot, that embolus circulating around, and it goes into your heart, goes out through your pulmonary arteries, into the pulmonary arterioles, and then gets stuck in a little arteriole in your lungs, you're going to lose blood flow to that whole chunk of lung. The opposite side of having a clot is not being able to have a clot. So bleeding disorders, thrombocytopenia, not enough platelets, impaired liver function. Most of those clotting plasma proteins like prothrombin activator and fibrinogen and plasmin, those are all produced by your liver. So if you can't produce those molecules, you're not going to clot properly. Hemophilia, as you may have heard of this, it's a genetic disorder or several genetic disorders. Usually most common in males because most of the types of hemophilia are found on the sex chromosomes. We'll talk about that in genetics. In any case, you don't clot very well and you tend to bleed a lot after an injury. And lastly is what's called, this is a big complicated thing, disseminated intravascular coagulation. Disseminated means spread out. Intravascular means within the blood vessels. Coagulation means, well, clotting. So certain conditions down here listed can cause this. If I'm type O and I get type AB blood, I'm going to probably have disseminated intravascular coagulation. I'm going to get agglutination and clumping of the blood throughout my body, but it can also happen from bacterial infections and certain pregnancy complications. Last slide, I promise. If you lose some blood, you might need some blood back, right? And that's what a transfusion is. You can either do it to restore just some volume or components of the blood itself. If it's just volume, you're usually going to get plasma or like a Ringer's solution, an isotonic solution that may include some plasma proteins or it may not. You can transfuse red blood cells, whether as whole blood. So I could take a sample of my blood out, give it to somebody else if they were matching, and they would be able to just assimilate that blood almost as their own. Or you can dehydrate it, pull out the plasma, use that for something else, and pack the cells for like maximum efficiency. Now the problem is that all of your cells have molecules on their surface, and these molecules fall into a class of molecules called antigens, in that they can provoke an immune response. Specifically, the antigens on your blood that we're talking about now are called agglutinogens because they generate GEN, agglutination, which is clotting or clumping. Type A, if you have type A agglutinogen on your blood, you're type A blood. If you've got a B agglutinogen on your blood, you're type B blood. If you've got A and B, you're type AB. If you got nothing, you got type O. So that I'm O negative, which means I don't have the A or B agglutinogen, and I also, because I'm negative, means I don't have the RH factor. RH factor is a completely separate gene. If you have the gene that causes that molecule to be deposited on your bloodstreams, you're RH positive. If you have a gene version that doesn't cause it to be deposited, you're RH negative. So I'm O negative, which means no A, no B, no RH. Type AB positive has all of them, so it can receive any blood type. If you were type AB positive, you can get A blood, B blood, O blood, positive blood, negative blood, any of the types. So you're called the universal recipient. By the way, I point to the lab videos and PowerPoints for more detail on this because I'm not going to go into as much here. you still have to know what's going on. So I provided that information in the lab video. Make sure you understand it. Type O negative, myself, have no antigens on there, none of these anyway, and therefore I can donate to all blood types. So I don't have A, B, or the Rh factor, so if you're a recipient of my blood, there's nothing on there that's new to you. The lack of something isn't something. Therefore, my blood's called the universal donor. However, on the flip side, I can only receive whole blood from an O negative person. Anybody O positive, A negative has something that I don't have, therefore I'd have an adverse reaction. And then quickly here at the end, when they take your blood at the doctor and they do a full blood work, let's say if they're going to do full blood work on a yearly physical, this can help determine if you have an illness and what may be causing it. So as just a preventative diagnostic measure, or as as a sort of a observational test or a diagnostic test, you can count the different types of white blood cells. So if you've got lots of lymphocytes, that may lead to one type of disease. If you've got lots of eosinophils, another. If you've got lots or very few, let's say, monocytes, that's another. You can count your platelets. You can count all of the blood, how much red blood cells you have compared to how many white blood cells, and so on. And then chemically, you can look at what's called a complete metabolic panel, or CMP, which is going to measure the molecules that are in the plasma. And you'll probably end up, if you get full blood work, they're probably going to look at all this stuff, right? You'll get back what your triglycerides are and what your cholesterol is and this and that and glucose and so on. Anyway, that's it. That's the last video for the lecture on Chapter 17, the blood. Study hard. Take down these notes.